So you can see me and hear me, and I would like to say welcome to everybody, and thank you very much for allowing me to be part of this school, which I have already enjoyed, and I know how difficult it is to make this online. I was organizing another international training school less than actually 2 months ago, and of course, it's never the same as a physical meeting, which we thought we would have in Spain in this case, but we will all do our best to enjoy this. And I start with this picture of spring or early summer, which we are not quite having, but we look forward to having on the west coast of Sweden in Gothenburg. And just to connect to a very nice introductory part of his introductory lecture, I refer to you, Alex, when you talked about brains and computers. I must say I very much share this view, and I want to build on it a little bit, because if we think of our brain, we should think of at least two immensely complex systems: the network of neurons and the network of astrocytes, on which we still have a very imperfect knowledge. Now, I completely agree that if we compare brain to existing supercomputers, there is essentially no comparison, because it's quantitatively something completely different. You know, our brain is so vastly superior, but there is also a distinction on a qualitative level, if you wish, because in contrast to your computer, which you buy with certain defined hardware, our hardware is profoundly and continuously changing. So as you listen to me, you are making billions of new connections, and many of them will be lost, but hopefully not all of them, and some of them will be translated through the process of memory consolidation into other connections, and some of them will really stay. And this plasticity potential is immensely important in health, but it is also important in a disease context. It's also important in aging, in particular, if we want to think of a healthy aging. And to make another connection to the talk, opening talk by Alex, I would like to say that those modalities which were touched upon as important for astrocyte function, they are of course very much important for the neuroplasticity, because of course neuroplasticity and astrocytes are also interconnected. And when I say those modalities, I refer again to enriched environment, to physical activity, to calorie restriction. So since we are a school, to really concentrate on another topic which actually was brought up already by Alex as well as Elena. So if we talk about a marker, gliofibrillary acidic protein, which neuropathologists have used for decades now, we should appreciate the benefits, but we should also appreciate the reservations. Now, one is at the level of number of astrocytes which are GFAP positive, which is of course disease specific, in a normal situation, region specific, and as was pointed out, even though the vast majority of astrocytes are actually GFAP positive, the levels can be sometimes very low, which of course in the limited dynamic range of immunohistochemical assays generates its challenges. Now, when we look at the other aspects of that, and that is what can we see by using GFAP immunostaining, and again, most of my clinical colleagues would actually always see astrocytes in that sense, because when we look at the histopathological samples, that's actually GFAP immunostaining. So that is what you have on the left, and you are very familiar with. You can see these star-like profiles. Now, should we call them astrocytes? No. This is really visualization, as was correctly pointed out here, of part of the cytoskeleton. I can be, of course, much more specific by saying that these are bundles of intermediate filaments, which we also sometimes call nanofilaments. Now, you appreciate the importance of this if you look at the panel in the middle, because what you see was an astrocyte randomly picked which we filled with a dye and did 3D reconstruction, and then you can appreciate this complex, bushy-like appearance with many processes. And in fact, if I should give you a number, the number would be 15. You visualize something like 15% of the volume of a single astrocyte by using GFAP immunohistochemistry, and I think that should be one of the take-home messages. Now, you all know, because this is Astroglia school, about the physiological functions of astrocytes, so I will not even repeat them. But I would like to say that in many CNS pathologies, almost all CNS pathologies, astrocytes respond. And this can be trauma, stroke, neurodegenerative diseases, as we most recently heard from Isidro. So they respond by something we call reactive astrogliosis, and this concept is perfectly comparable with compatible with this heterogeneity of responses which Alex brought up, and which also would include astrocyte atrophy. Now, when it comes to reactive astrogliosis, it has number of many genes get differentially expressed, as you have also heard by listening to Elena already. One of the hallmarks would be upregulation of the components of the cytoskeleton, in particular intermediate filaments, GFAP, nestin, vimentin, and there is process hypertrophy of reactive astrocytes. Now, reactive gliosis is, as we always often try to view it, as a two-edged sword. It can be positive, and in many contexts it is, helping to handle acute stress, limiting the damage to the brain tissue, restoring homeostasis. But the key word here is something which already has appeared, and that is context-specific response, or disease-specific if we view it purely from a clinical standpoint. Because in some contexts, reactive gliosis can be maladaptive, and if it persists, can also cause problems, hampering many aspects of neurodegeneration. Now, of course, our improving molecular understanding makes it closer and closer the possibility to ultimately interfere therapeutically targeting astrocytes. Now, another point which was raised here, and I thought I could really add to a very good foundation which was already presented here, and that is what really happens in a morphological sense to astrocytes when they become reactive. Well, we did this work together with Mark Ellisman in La Jolla, in San Diego, at the National Imaging Center, after they had very nicely described the normal and developmental situation, which would be similar to what you see in this picture on the left, namely that astrocytes they exist in tile domain with very limited gap region of overlap, which of course is the gap junctional communication, here shown in yellow. Now, our interest here was something which most likely interests you too. What happens when astrocytes become reactive? We used two injury models in the brain, very different ones, and we got the same outcome from both. And what we found that actually reactive astrocytes, they pretty much stay within their tile domains. They don't increase the volume of the tissue they could access through their processes. But what really happens is that the thickness of the main cellular processes increases, and as a consequence of that, you can follow the bundles of intermediate filaments for a longer distance. So this gives, when you do the imaging without this state-of-the-art reconstruction after dye filling, if you just concentrate on the intermediate filament proteins, intermediate filaments, then you of course have an idea, a false impression that there was a true cell hypertrophy, that the cells became bigger. They don't, and of course it's very easy to understand why. It's very difficult to imagine that in a defined volume the connection between the astrocytes would persist when they would increase their volume. So of course, again, as you can see here, the number of processes, the main cellular processes measured certain distance from the cell soma would be increased in reactive astrocytes once more, proving that we are talking about process hypertrophy. Because this is a school, I'm sure I will not offend you by this picture from a textbook. When we talk about cytoskeleton, then we talk about actin filaments, known also as microfilaments, microtubules, and intermediate filaments, known sometimes as nanofilaments. Now, everybody in Sweden at the level of gymnasium learns about actin filaments and microtubules, and typically students are very happy because they think microtubules are highways on which different trains, meaning molecular motors such as kinesin or dyneins would transport different cargo. But intermediate filaments, they don't have even polarity. So they function in a different way. It's still, to some extent, enigmatic. The bundles of filaments would disperse upon activation in a given cell type. Intermediate filaments are present in all cells, apart from red blood cells, which are not cells, and they really react upon activation. And we view them more and more as a crisis command center of respective cell types, of course, including astrocytes. So astrocytes have three, actually we can these days say 4 intermediate filament proteins: GFAP, vimentin, nestin, and cinnamin, in various proportions. The stoichiometry is, of course, very much determined by developmental stage, subtypes of astrocytes, the regions, and would very much increase in connection with defined pathology. So the functions of intermediate filaments, or the connections with the cellular functions, if you wish, few are known. One of them is cell motility. Here, if you compare, and this is already important to make one point, GFAP knockout, which we made many years ago, was a useful tool, but there were two major challenges. One challenge was that it gets partially compensated by vimentin, another intermediate filament protein. And the second was that, as I already mentioned, the stress proteins, very little is seen in a normal situation. That was actually an advantage, in fact, with respect to understanding the role of intermediate filaments in astrocyte activation, in reactive gliosis, and also to really understand some aspects of disease-specific reactive gliosis. So you can see the same dose effect, if you wish, the partial depletion, meaning GFAP null or vimentin null primary astrocytes in vitro, they would have somewhat decreased motility, but you would see that much more profoundly expressed in the absence of both GFAP and vimentin. Now, if you break down the subpopulations of cells depending on their migratory speed, in a normal situation of primary astrocytes, you get something very close to normal distribution, and you can see partial and more pronounced depletion of fast moving subpopulations of astrocytes when you genetically ablate either GFAP or vimentin, or of course with a more prominent effect when you deplete GFAP and vimentin, which results in no intermediate filaments being in those cells. And this is what happens when you remove nestin. You should look at this picture, the left bottom, the other way around, because it's a speed of a closure of an artificial wound in the dish. So you can see that nestin astrocytes, they really lag behind, and there is something like 20% reduction in the scratch wound assay closure at day two. Now, when you think cytoskeleton, and when you think skeleton as such, you of course think mechanical, you think resilience. I think there is a difference if we ask a general practitioner what the skeleton does. It would probably be the mechanical role plus the bone marrow inside it. The general population would say structural support. So is there any structural support connected to intermediate filaments? These experiments were done together with our colleagues and friends at University of Leipzig, Andreas Reichenbach, Christian Franze, who now runs his own lab very successfully at the University of Cambridge. And we have asked whether or not intermediate filaments are connected with increased stiffness. And we turn to these cells, which I will actually introduce in a few moments, and they are the Miller cells of the retina. Just remember the term, those of you who don't know them. We challenge the retinas, which are of course part of eyes, by retinal ischemia. And then you can see in the wild type system, when you then isolate those Miller cells and you expose them to scanning force microscopy to assess their viscoelastic properties, you would clearly see that both in the region of end feet and inner processes, the stiffness would increase with increased production of intermediate filaments. And of course, when there are no intermediate filaments, the basal stiffness is not affected, but there is no increase whatsoever in the stiffness of Miller cells upon or after they were isolated from the ischemically challenged retinas. So this established that clearly the viscoelastic properties, if you wish, in this particular case, the stiffness of reactive glia depends on intermediate filaments. And here's the promised picture, and I should really give a credit to Andreas Reichenbach and Christian Franze and other colleagues, because they found a new piece of physiology which will soon appear in textbooks, and that is very rare to happen these days, namely finding that Miller cells, they act as fiber optic cables, conveying light to the receptor cells. So during development, they guide neurons their final destination. Some of them also turn into neurons themselves, but there are few places throughout the central nervous system where radial glia persist, and one of them would be Miller cells, others would be Bergman glia. Now, I will stay with Miller cells for one more minute, and this gives you an idea that when one attenuates reactive gliosis in this particular genetic way, genetically ablating GFAP and vimentin, so no intermediate filaments in those mice, what really happens is that you can see in this model of retinal detachment in the mutant mice, again, attenuated reactive gliosis, there are clear signs of attenuation of their responses, such as reduced ERK or CFOS activation. There is also a reduced level of monocyte chemoattractant protein 1. And what is interesting, and I want to draw your attention to that, look here on the right, there is reduced apoptotic death of photoreceptor cells, which of course then result in a less pronounced retinal damage. So in this case, to reduce or attenuate astrocyte reactivity is a good thing, because you can easily see here that we have more of the thickness of the retina protected. So you can see some examples of this sort. I would like to bring your attention to this collaborative study published some time ago together with our colleagues at Harvard Medical School, Dongfang Chen's group. And we asked the question which has been around had been around for a long time, and that is whether or not the lack of success or very limited success connected to various transplants in the central nervous system could be caused by induced, iatrogenically induced reactive gliosis. And again, we turn to the retina as a very nice part of the central nervous system, which is nicely experimentally approachable. And we used a model which had been long established before, and these are the retinal transplants, essentially transplanting cells from dissociated retinas. Of course, to be able to follow them, we took them from animals expressing enhanced green fluorescein protein, and you can see that nothing happens in wild-type recipients. Now, what happens in mice in which we genetically attenuated reactive gliosis by removing GFAP and vimentin, you can see on this panel these cells derived from the grafted cells. They really repopulated the retinas. They established themselves very nicely. You can see this difference compared to the wild-type recipients with normal reactive gliosis, and they even send axon-like processes all the way into the optic nerve. So clearly attenuation of reactive gliosis increased very substantially the permissiveness of the environment to integrate neurografts, and we then moved this also to the brain, and these publications are available. Now, moving to the truly disease context. Now, stroke, of course, as you know very well, stroke is extremely heterogenic unit. The term is derived from the fact that there is a strike, there is a very profound occasion which is part of this drama-like situation. And there are a number of reasons, but the two main ones would be ischemic stroke, where there is a physical obstruction within the vascular system. And then clinically, we distinguish thrombotic stroke, where this obstruction would build in place, and embolic stroke, where the plaque which obstructs the vessel would get there from elsewhere. So the other category is hemorrhagic stroke, which is a consequence of a rupture of a blood vessel. So in this case, we ask whether or not attenuating genetically reactive gliosis in ischemic stroke, and the ischemic stroke was induced by transection of middle cerebral artery, which is the very artery which becomes very often obstructed in ischemic stroke in a human context. And you can see very well that already at the level of infarction volume, we saw that there was increased tissue loss after stroke in mice with attenuated reactive gliosis. These were two different models, the same outcome. We have meanwhile understood some of the mechanisms, and there was a series of papers, and I will just point to one example, that reactive astrocytes at the acute post-ischemic stroke are very much important for reactive oxygen species elimination, and this process is impaired in astrocytes with attenuated reactive gliosis, and of course affects the degree negatively of neuroprotection. Now, I'm going to share with you something which is unpublished, but it is so exciting that I thought it's worth to share it with you. In collaboration with Matthias Höhn and Markus Aswend from Max Planck Institute in Cologne, we asked a question whether or not after stroke, reactive gliosis affects the neuronal connectivity, because you can imagine that a stroke is a devastating disease, which would of course disrupt many connections in the brain, and then there, due to the plasticity of the brain, there would be a certain degree of recovery through the whole variety of mechanisms. We have written few reviews on the topic. You are all familiar with remapping, for example, but there will be number of connections, local and global, changed. Of course, you know that unless one dies in the acute stage, there is a substantial degree of improvement among stroke patients, which we can therapeutically enhance by rehabilitative interventions. So we have subjected mice to photothrombotic stroke, which is another stroke model in this context used on purpose to limit the ischemic penumbra, because we were primarily looking at the neuroplasticity responses. And we have seen that immediately after stroke, those connectivity responses, which are the newly built on the building, and the lost, in particular, the lost connections, there was no difference between mice with and without attenuated reactive gliosis. However, at a later stage, we saw aberrant restoration and sensory motor neuronal connectivity, which was also connected with increased plasticity, both glioplasticity and axonal plasticity, which we saw a month after stroke. So this shows that reactive gliosis is important for normal neuronal connectivity after stroke, and we are getting some mechanistic insight into this. The interesting twist in the story is that, as you know very well, a stroke patient, even if he or she would be very lucky and would normalize functionally, such a person would still have a problem until the end of his or her life. Why? Because there is something we call secondary neurodegeneration. So if you look, for example, at the annual average loss of brain tissue after stroke, as one ages, there would be a clear increase over matched controls. And this, when we look at the secondary neurodegeneration in the thalamus, in this case assessed as amyloid accumulation, we also see that having normally responding astrocytes, meaning the wild type situation is better than astrocytes with attenuated reactive gliosis when they are deprived of GFAP and vimentin. Now, I would like to revisit this kind of theme which I already prepared when I talked about the distinction between the ischemic and hemorrhagic stroke. So again, hemorrhagic stroke would be in sharp contrast to ischemic stroke, bleeding. And of course, that bleeding would generate major problems, because you will also have enormous vasogenic edema. There will be, which is of course a major problem also in a situation when the volume of the brain cannot expand without very heavy cost, and that is increased intracranial pressure, which of course in many cases can be a direct cause of death. So in this case, we use collagenase model. I should give a credit to my postdoctoral fellow who comes from Spain, Maria Petraza, and this is in this model, collagenase would disrupt the blood vessels, and that would lead to a bleeding. And we ask a very simple question: genetic attenuation of reactive gliosis, GFAP, vimentin. Now, compared wild-type mice, would we see any difference? So, to get a nice baseline, we were happy to see that the hematoma size was comparable. Now, looking at the compiled neurological score, both the normal, which means a pre-stroke score, or any normal adult mouse would have 21, just for you to have a proper comparison. So this is what really happens in wild-type mice, 24 hours after, 72 hours after, and then suddenly you see something striking, don't you? It's exactly the opposite to ischemic stroke. In this case, to genetically attenuate reactive gliosis is a good thing, because GFAP, vimentin, null mice are doing better. And it's actually quite profound. And then, of course, when we follow the whole thing, we realize that they have very substantially, there is a trend at 24 hours after stroke, but 72 hours later, there is very substantial reduction in edema. And as you know, it's mostly vasogenic edema, responsible for edema after hemorrhagic stroke. And then, of course, edema brings you to the water flow controls, and when we looked at the aquaporin 4, the main water channel positivity, we saw that there it was very much increased in GFAP vimentin null mice, and even more so when we looked into a subcompartment of aquaporin 4, namely the estrovascular subcompartment, where astrocyte processes are in direct opposition with endothelial cells. So again, to attenuate reactive gliosis in hemorrhagic stroke leads to better functional recovery. I didn't show you data, but there is actually reduction in neuroinflammation. There is a high, profound reduction in post-stroke edema, and as I already said, there is a major increase in vessel-associated aquaporin 4, which, as I just said, is the principal water channel of astrocytes, and we are also seeing increased dynamics of aquaporin 4 response after stroke. What does this show? It shows exactly what you have already heard in Alexis and Elena's presentations, namely that context matters. Context matters. And because there was a lot about Alzheimer's disease here, I included this story because I completely agree with Elena and with her statement that the role of reactive astrocytes in Alzheimer's disease is unclear, and it is very, very important to decipher. Now, you can see the amyloid plaques in blue, and you can see quite profound presence of microglia as well as activated astrocytes around those plaques. And now I will refer to the work which we did with a wonderful colleague and a friend, Yinwu Li from Washington University in St. Louis, and we ask a very simple question, and that is, would genetic attenuation of reactive gliosis in this particular way, ablation of GFAP and vimentin, which means no intermediate filaments, would it really make any difference? So this was our Alzheimer's disease model, the combination of the Swedish APP mutation with prestanolin, and again the mice which I already nicely introduced to you. Already at the level of quantifying amyloid plaques, you can see in particular 8 months to 12 months, you see a clear difference. Increased amyloid plaque load in GFAP vimentin null mice. Now we wanted to visualize astrocytes, but we didn't really want to do it by injecting individual cells. So this was related to one of the questions which Alex answered before lunch, how to do that. So this was a viral construct, and I will show it in this a rather nice way. So we injected it in the mouse cortex at 4 different places, at 2 different depths. And if you see at this low magnification picture, you can appreciate that quite a portion of the brain cortex would turn and did turn green. So if we zoom in, you can already see the phenotype very nicely, because this is what happens in Alzheimer's disease mice on the background of GFAP vimentin normal. So again, you see amyloid plaques in blue, you can see astrocytes in the direct vicinity of these plaques, but then you also see something much less pronounced, don't you, in mice with attenuated reactive gliosis. And if we zoom at this, and let's do it now, you can see that in a normal situation, there are astrocytes in opposition to these plaques, very close opposition. So what happens with attenuated reactive gliosis in that situation is that we have the immediate vicinity of amyloid plaques almost depleted of astrocyte processes, and this needs to be quantified. You can see a comparable number of astrocytes, and you can see here very clearly that the astrocyte overlap with amyloid plaques is very, very much decreased. Well, now you would then say, okay, but does it matter? Well, we quantified the extent of neurite dystrophy, and indeed we found that it was more prominent in Alzheimer's disease mice on the background of attenuated reactive gliosis, which allows to make a conclusion that reactive gliosis in Alzheimer's disease might have positive roles. Of course, we should always view this critically because, as I already indicated, this way to manipulate or attenuate reactive gliosis is really one of many, and we are realizing this more and more. The lab headed by my wife, Professor Marcela Pekna, they are looking in the ways how through the complement system signaling one can modulate reactive gliosis, and there are interesting parallel opportunities along that track. Now, I would like to actually conclude this segment by saying that clearly Alzheimer's disease and this particular model suggested that in that context reactive gliosis is positive. We have completely opposite data for amyotrophic lateral sclerosis, which I do not have time to share with you today, but I'm ready to discuss it. I would like to shed some light on nestin, which is another intermediate filament in filament protein in astrocytes, which is expressed early on during development and at selective places throughout the central nervous system, and is a very prominent marker of reactive astrocytes. And neurogenesis was already kind of introduced gently by Alex, so I just want to say that it's very obvious that it's extremely active in prenatal period. The whole brain and spinal cord are derived from a neurotube, which originally is only a single cell layer thin. Then in adult mammalian brain, it persists at a very, very reduced rate in two areas, hippocampus and subventricular zone, that it is still relatively prominent in rodents. In higher mammals, such as ourselves, it's still a question to what extent adult neurogenesis has a functional significance and how much of it we have. In any case, adult neurogenesis is a very tightly controlled process, and as you can imagine, would depend both on the intrinsic properties of the stem cells, neurostem cells, and also on the environment, which we can call neurogenic niche, and which consists of glial cells and, of course, of surrounding blood vessels. I think it's very important because due to the fantastic work of Rusty Gage and his colleagues at Salk Institute in La Jolla, there has been a little bit one-sided view of neurogenesis, like neurogenesis means better capacity to remember new things, and that is correct. But everything comes at a price, and there are two sides, because you can imagine that hippocampal neurogenesis leads to reorganization of the circuitry in the hippocampus, and therefore it is linked both to the formation of new memories, and because it's a structural reorganization, as I have just said, is connected with the loss of previously acquired memories. And we used nestin knockout mice, and we asked the question to what extent nestin really plays a role in either neurostem cells or in astrocytes, because people in the neurostem cell camp would, of course, expect a role there. These mice, I can reveal, they live quite normal lives, so that means that there is no profound involvement, at least not the one which would not be compensated, of nestin in prenatal neurogenesis. Now, when we looked at neurostem cell proliferation and the neuronal precursors, the double cortin positive cells, using the BRDU in vivo labeling, which means that this is an analog which gets integrated into the DNA of those dividing cells, and you can, of course, quantify it. You can see that nestin doesn't affect neurostem cell proliferation. Does it affect neurostem cell differentiation? The answer is yes. If it was not the case, I wouldn't have shared this picture with you. So you can see that 5 weeks after this injection, there is an increased number of neurons in the hippocampus, one of the adult neurogenic niches, and you can see it by both using nu-N or calbinding, the two well-established markers of neurons. Now, we wanted to ask the question which some of you who will listen to my presentation already have, and that is it neurostem intrinsic property or is it caused by the niche? And for that, we took advantage of a number of in vitro systems, and by doing that, we first established that the absence of nestin didn't affect any aspect of neurostem cell proliferation or differentiation or migration towards a gradient of SDF1 alpha and other parameters. So it didn't seem to be any effect on the neurostem cells. But then by turning into both systems of astrocyte neurostem cell co-cultures and the system of neurospheres and neurosphere dissociated cells, we were able to show that the absence of nestin indeed affects neuronal differentiation. There is clearly the same increase, actually even more prominent than the one seen in vivo. So now through the co-culture experiments, we were, by using pre-labeled neurostem cells and co-culturing them with either nestin null or wild-type astrocytes, we were able to establish that this was not, and now it starts to be interesting, very much for the astrocyte field, it was not the neurostem cell intrinsic effect, but it was the effect of a niche. Namely that nestin null astrocytes, they provide a more pro-neurogenic environment for those neurostem cells. And then through additional experiments such as using mixed cells or culturing them the way that they couldn't have physical contact, we were able to establish that this pro-neurogenic environment of nestin null astrocytes requires contact between individual cells. And finally, we were able to track it down to notch signaling from astrocytes, from astrocytes as signal sending cells to neurostem cells as signal receiving cells. And we were able to show that actually nestin null astrocytes were unable to sustain over time the notch activation of co-cultured neurostem cells. And this was in a collaborative arm together with Robert Zorec from the University of Ljubljana in Slovenia. We were able to track it down to an altered vesicle trafficking. So a connection between intermediate filaments and vesicle trafficking. It led to the whole range of studies. In this case, the vesicles containing the principal ligand of the notch signaling system, which is JAGIT1. And you can see that in particular, the REP5A vesicular endosomal compartment was very much affected. And in a recently published collaborative study, again with Robert Zorec, we were able to show that nestin affects the fusion pore dynamics in astrocytes, which affects the size of the vesicles which interact with the plasma lemma, the fusion pore geometry and kinetics, and also is important for purinergic calcium signaling and the cytoplasmic vesicle trafficking. And those of you who listen to me carefully would say, okay, but there is one issue here, because nestin then must be expressed in a subpopulation of normal astrocytes, unchallenged astrocytes. I said it, but it was not really part of common knowledge, because otherwise how would you expect that effect on the neurogenic niche? And the answer is definitely yes. You can see here that there are astrocytes in the adult in the neurogenic niches which are nestin positive. Of course, nestin is absent in the nestin knockout. It does not affect formation of the intermediate filaments. You can see that GFAP and vimentin form intermediate filaments in both in vivo and in vitro in the absence of nestin. Now, I would like to conclude this all the way. I told you on the initial slide that if we think of the reorganization of the hippocampus, and if nestin enhances that through increased neurogenesis, you would expect effect on cognition. And indeed, in two recent studies, we have seen that there is an enhanced reversal place learning and memory extinction. So they both more easily forget what they have learned and are better learners. So I would like to conclude this part by saying that nestin negatively controls neurogenesis, and that is not neurostem cell intrinsic, but on the contrary, it's through the environment, namely the astrocyte niche. It's required for normal activation of Notch signaling and the signal transmission from astrocytes to neurostem cells. Nestin plays a role specifically in controlling the segregation of the principal ligand of Notch, namely JAGIT1, into very distinct vesicular compartments, and these are specifically the early and recycling endosomes. Nestin is required or plays a role in normal astrocyte migration, and as I just said, synaptic integration of neurons into existing hippocampal circuits, which does happen in adult mammalian niche, then facilitates both learning, that's a gain, and forgetting of previously stored information because of the rearrangement. And indeed, we do see enhanced reversal place learning and facilitated forgetting in nestin null mice, which might reflect this increased survival of hippocampal neurons. And now in the last segment of my talk, I would like to turn more molecular. There were discussions about the GFAP presence in astrocytes. I just want to give you this feeling of the range of very wide range. This is at the level of messenger RNA molecules of GFAP. And these are freshly isolated cells. So no culture step, freshly isolated cells from either injured hippocampus or the contralesional or control. And this was done 4 days following entorhinal cortex lesions, which, as many of you know, would lead to partial deafferentation of the hippocampus, specifically the outer molecular layer. And of course, you can see that in a control, which means uninjured situation, the difference, this is relative gene expression in the levels of GFAP between low and high expressors would be more than 30-fold, but it would become more than 3,000-fold. So this was also the range, which of course is outside the dynamic range for any detection we can do with antibodies. So we have used single-cell gene expression profiling, which of course is a very useful method to identify subpopulations of cells based on their molecular signature, and typically an expression of a given gene shows log normal features. So if this is a logarithmic axis, you would have a normal distribution. Vimentin is one of very few exceptions. You can see low vimentin expressors in red and high vimentin expressors in green, and you can actually follow them when you look at the expression of other genes. And this allowed us to really identify in primary cultures of astrocytes these two distinct subpopulations where the low vimentin expressors shared many molecular characteristics, as this principal component analysis shows with with neurostem cells, while the high vimentin expressors shared many molecular characteristics with reactive astrocytes. I just want to refer to one study which was also possible due to the ability to interrogate astrocytes on a single level. This was actually done originally in the mouse context, and that was finally went to clinical data, clinical material in humans. So when we used experimental injury of the kind I just described to you, we have seen a subpopulation of cells which, interestingly enough, were present only in the injured situation, here depicted as MA cells, which expressed both markers of astrocytes and markers of microglia. We don't know what these cells really do. We looked in the human situations, and we have seen that in patients with stroke, Alzheimer's disease, and also Lewy body dementia, we identify these dual identity cells, and there are now some other groups looking at them in the context of amyotrophic lateral sclerosis. And at the very end, I would like to share with you a story which would probably make many of us interested in GFAP again. And this is an isoform of GFAP. I will don't worry, I go to that picture back in a few moments. I just want to share with you a picture with which all molecular biologists like. So here is GFAP as you know it. It's alternatively spliced, so there are several isoforms, and these are the three additional players which I introduced to you: vimentin, nestin, cinnamon. You can read more about that in the review we wrote together with Ali Hall and published in Current Opinion of Cell Biology about the isoforms. Now, going to the very novel and unpublished data, GFAP delta is highly expressed upon injury. In fact, the expression is very much increased with a much bigger factor than that one of GFAP. So this is the same model which I already introduced to you, the efferentation of the hippocampus, partial one, after unilateral entorhinal cortex lesions. So I would like to draw your attention to this rather conceptual picture, because here, by looking at selected astrocyte markers, you recognize most of them. This one is GLT1, GLAST1, glutamate transporter. This one is GET3, which is GABA transporter, endothelin receptor B, azrin, glutamine synthase, SOX2, thrombospondin 4, vimentin, nestin, cinnamon, and these are additional genes, actin and C1q, within the complement pathway. And most of you recognized this favorite gene of Ben Barres, aldehyde dehydrogenase L1 isoform. So if you look at these genes, and here it's actually a subset for these pictures of these four tentative markers: aldehyde dehydrogenase L1, GFAP, GLT1, and GLAST1. And you look at the astrocytes isolated, direct isolation from uninjured brains, you can see that 43% of astrocytes would express all four markers, and in contrast, 31% would express only a given marker, one of these four. Now, the injury changes this quite dramatically, because then the astrocytes would kind of increase their molecular diversity, if you wish, get molecularly more specialized, because now only 20% of astrocytes would express all of these, but many more would express only a single one, in this case 58% compared to 31%. Now, I would like to bring up the point that, or make the point that GFAP delta is a powerful and very interesting classifier of astrocytes in the injured hippocampus, because if you look at the control situation, and here you have GFAP delta positive and GFAP delta negative situation, you see differences, of course, but they are not very profound. Now, In the injured context, suddenly, you see major differences. Now, this is very important to understand three things. Number one, on a binary level, GFAP delta expression, yes or no, it's roughly 50-50 in control and the injured situation. So there is no difference there. We're talking about the same size of those subpopulations. But as you are all familiar, or most of you, with the way how can quantify this. So the size of the circle gives you an idea about the percentage of cells positive for the given gene, while of course the color, it gives you an idea about the level of expression. So the darker the color, the higher the level. So you can see that suddenly GFAP delta in the injured situation can be used as a very powerful classifier. And if you look at this principal component analysis, you can see that actually the GFAP delta expressing astrocytes, they end up in largely distinct, not completely, but largely distinct crowds in the controlled and injured situation. Now, this is a very nice built-in control, because with entorhinal cortex lesions, we have a kind of in-between situation, and that's the contra lesional hippocampus. And you can see that for all these genes, essentially, we are looking at, there is a situation in between. If we look at control and injured with a contra-lesional being in between, having a dose response. Now, if you think of the single cell analysis, you should always appreciate the power at three levels. One level is binary. Of course, you can see a given cell expresses a given gene or does not, on-off situation. The other level is, of course, what is primarily meant for, and that is you quantify the level of expression. You can essentially get close to counting messenger RNA molecules. And the last one is the correlation. And that gives you, of course, additional possibilities to learn, for example, about probable common transcriptional control. And it's very interesting to see that all these genes which I introduced to you about 3 minutes ago, if we look at the correlation between their expression and the expression of GFAP delta in an injured situation, there is nothing. But suddenly, there are many of these, which there is actually not even a correlation between GFAP and GFAP delta. But in the injured situation, there are quite a number of correlations, including the strong ones with GFAP, aldehyde dehydrogenase L1, endothelin receptor B, and vimentin. So I think this is the last picture. This actually allowed us, even though this this was a planned candidate approach, this allows us, using these self-organizing maps, to identify 4 astrocyte clusters from these directly isolated astrocytes from either injured, uninjured, or contra-lesional hippocampus. The one in yellow, which with respect to the gene expression would correspond to non-reactive astrocytes. They are partially overlapping, but still assuming largely unique space. These, which are reactive astrocytes or correspond to reactive astrocytes, you can see the respective markers. Then there are cells which are either undifferentiated or dedifferentiated, which actually very often happens in conjunction with a trauma-like situation. And then you also see a cluster enriched for those dual identity cells, the cells which actually express also markers of microglia. We are not sure whether this could be, at least in part, a consequence of cell fusion. And the very last slide shows you how we are trying to move this into the clinic in a collaboration with Jan Mulder at Karolinska Institute. We are using tissue arrays from patients who died of stroke, Alzheimer's disease, or Lewy body dementia. And on this picture, which I would like to share with you, you can see that the GFAP delta expression is increased in the brains, not of all. Let's be fair, on average it is increased in Alzheimer's disease patients, but if you see and appreciate the data for individual patients, you are not, of course, surprised, because Alzheimer's disease is an extremely heterogeneous category defined in mostly in clinical sense. We know there are many diseases under that umbrella. It's similar as is the case with ALS, but you can see that in quite some cases of Alzheimer's disease, the expression of GFAP is increased, and you can really see those GFAP delta positive aggregates. You see them in age-matched controls, but nothing at the level of what can be seen in some patients. So I would like to conclude that reactive gliosis is context specific. We are learning by addressing the question of the role of reactive astrocytes in different diseases. We're learning about the complexity, and we see that not always nature follows the optimal path, which of course gives space for therapeutic interpretation and intervention. So we have quite recently started with some colleagues this EU joint program in rare diseases, which is one of the 22 rare disease programs financed by EU. Alexander, because as Alex so nicely presented, Alexander disease is the first monogenic disease known to originate in astrocytes, and it's caused by mutations in GFAP. So the proper term is the astrocyte nanofilament system in Alexander disease, from molecules to function, uncovering new leads for therapy. And with this, I would like to acknowledge people who are involved in this work, both from my laboratory at Clinical Neuroscience in Gothenburg, and number of very pleasant collaboration with a group of my wife and Turgut Tatlisumak, Jan Mulder at Karolinska, I already mentioned that, Matthias Hön, and Marcus Asvend, the neuronal connectivity, Ellie Hall from University of Utrecht, Christian Franze from Cambridge, Tata, which are our collaborators when it comes to single cell gene expression profiling, and the same applies to Mikhail Kubista. And as I already mentioned, Yinmu Li, the Alzheimer's disease project at the University of Washington, and I also mentioned Robert Zorets and Cecilia Salgren from Åbo in Finland, and I want to thank you for your attention.
Okay, thank you very much. Thank you very much for your presentation. A couple of questions. There is one very you can maybe your question.
I see. I hear it. I don't know if it's on you, but yeah. So I actually have kind of not so. Yeah. Compared to the rest of your talk, it doesn't seem so important, maybe. But I have a question about your in vitro system, where you measure the stiffness of these Müller cells. I assume you did this on plastics, so just in normal well plates.
Thank you for the question. Actually, the question is brilliant because it helps me to clarify that. Of course, if the cells were lying on plastic, the interaction between the cells and plastic would obscure the readout. Yeah. So these are optical tweezers. So you actually have them, so to say, suspended in space, and you actually see my hands, right? So that is a very well established technique how you can measure the stiffness. So again, there is no plastic and no substrate involved.
So it's like
So
in vitro.
they are isolated. You know, they are directly isolated from the retina's Miller cells, which then are isolated either from unchallenged retinas or from retinas which were exposed to hypoxic injury. And hypoxic injury would increase their stiffness, but if they don't have intermediate filaments, it won't happen. Hence, the stiffness is determined by the intermediate filament system.
And is it measured with force?
Now I couldn't hear you.
Sorry, is it measured? Okay.
Yes, of course. It's atomic force microscopy, and you can this was the optical tweezers, but atomic force microscopy can very much be involved. I'm happy to either discuss it by email, or you can very easily find the paper because this was published. Reichenbach, Pekny, you find few papers. This would be easy to identify.
Okay, thanks very much.
Thank you.
Thank you very much. There is another question. My colleague.
Yes. Thank you, Milos.
Thank you.
You show a lot of information. Well, I'm listening to time. Okay, you mentioned astrocyte reactivity and how it's context dependent and all that, but I remember, for example, this paper from Shane Lidelow and Ben Barres, where they, of course, like Elena said before, is maybe too simplistic, right? But they describe three main microglia factors that stimulate astrocytes in this way to induce the A1 astrocyte reactivity profile, or whatever you want to call it. How would you do you have any insights about how microglia could also affect the reactivity that you are kind of like explaining today?
So I was saying this is a great question because I can briefly comment on it at several levels. So first of all, I would say let's read this consensus paper in Nature Neuroscience, which will make us more critical towards the A1, A2 classification. So your
not more critical, just forget it. I mean, stop thinking about.
Yeah, that is what it was a euphemism, but that was my message. Thank you, Alex. But the question is brilliant because you are essentially pointing to the interplay between astrocytes and microglia. I haven't mentioned that, but if you look at the paper which I briefly commented on, and I of course showed just highlights on attenuation of reactive gliosis in Alzheimer's disease, then there was immediately a compensatory action from a microglia. They moved into the vicinity of amyloid plaques, and there is from many groups there is emerging molecular picture about what is the response from the other cell type when you manipulate microglia, what comes from astrocytes, and the other way around. So I think that the next school we should have a segment on astrocyte microglia interactions. It's a very incomplete picture, but it is exactly the kind of reasoning we are aligned with. Yes.
Yeah, so you always or in general gliosis right? So my question is, you can find also the same kind of specific reactivity, a behavioral challenge. For example, cognitive task. You see that the solving maze for days, something like that. And And if you can specific activity also, that could be protected for injury.
You know, Edgar, you are bringing up conceptually something very important, because we should think of astrocytes are always active, but they are active to varying degree and in different ways. So astrocyte, that's why we are skeptical to the term astrocyte activation, because it can be interpreted in a physiological way, or some people use it in a pathophysiological way. We all use it in some ways. We haven't fully decided in the field on the terminology, but the point which you are bringing up is very important, because there are astrocytes in parts of the brain which, for example, have very much increased in a relative sense to other locations the amount of GFAP and other prospective markers. So yes, there is a continuum between astrocyte activation and reactivity in the pathological context. And as we have published in a few reviews with Alex and with Mike Sofroniev, even reactive gliosis is very graded, right? I mean, the glioscar is the most prominent part, but in many cases, it's a very much reversible situation, which makes it even closer to the physiological response. Because, for example, if you make a subtle injury in the brain, then nestin would go up, and then in 2 weeks you won't recognize it, and you would have problems identifying those astrocytes. Magdalena Goetz, she wrote a paper where they were looking at these extremely subtle injuries. So there is a continuum, and it is the region specificity is very important, because the given insult in the spinal cord. Now, when I compare this relatively subtle injury I just presented and compare it with the spinal cord, then Jonas Frisen's data from Karolinska Institute show that that injury in the spinal cord would actually stay until the astrocyte reactivity will stay until the end of mouse life. And we have many clinical correlations to that, because as many of you know, spinal cord injury, for example, leads to persistent reactive gliosis, and there is gradual expansion of the injury and the cavity. So as we always say, clinically, the rehabilitation works up the hill. So I think that we should appreciate, Edgar, this continuum which you have been pointing to, and should definitely not disregard what is physiological and where the physiological challenge ends and the pathological challenge starts. It's very difficult, because if we define disease as a homeostatic imbalance, you really see, you should see that as a continuum.
Okay, thank you very much. There is no any other fellow organizer, so I think we can close today. We hope to see all of you tomorrow for the second session.